This is episode 277 of the Stem Cell Podcast, Lung Development, with Dr. Behzad Yeganeh. Hello, everybody. We are Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. And if you're enjoying the podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests for the show. Today, we have something a bit new along those lines. We got a guest on the show who the producers met at the ISSCR this past summer. And he was invited onto the show. And today we will be talking to Dr. Behzad Yeganeh. He's from the University of Ottawa. He's here to talk about his research on the mechanisms of lung development and neonatal lung diseases. We've also got our usual roundup of recent highlights and stem cell news. That's coming up.
But first, this week, we want to introduce pulmonary cell news, covering everything lung. From COPD to cystic fibrosis to cancer, Pulmonary Cell News keeps readers current with the latest news, research, policy, events, and jobs relevant to the lung research community. Check out Pulmonary Cell News and the rest of Stem Cell scientific newsletters at www.pulmonarycellnews.com.
All right, you guys, I'm kicking off the roundup today with what I think is like a true watershed in terms of like full circle application of human pluripotent stem cells. And I'll elaborate that on a minute. This is a story that the authorship in here is kind of confusing, quite frankly. It's a cell story that is the lead contact here is Hong-Ki Deng, and I think rightfully, because this is really the culmination of many years, decades of work that was coming out of his lab for this specific purpose, like all converging on this specific purpose. But there's another last author, Zongyang Shen, and 11 co-first authors. So this also clearly a culmination of a lot of individuals' work and effort. But with that kind of authorship, you'd think it was like a clinical trial or something, and it was. Don't get me wrong, but you'd think it'd be like expansive in hundreds of patients or many patients at least. But this was just one patient we're talking about. But that's really the key and the anchor of this story. It's the application of these chemically induced pluripotent stem cells in a patient with type 1 diabetes. All right, so just going back a bit, the first major milestone out of the Deng lab here was the generation of these chemically induced pluripotent stem cells, right, which are fundamentally, because there's no transgenesis, made with a complicated but straightforward, repeatable method. These are human pluripotent stem cells that are truly, I would say, optimally suitable for therapeutic applications. I would say that the most likely for the FDA to sign off on. So that's one big deal that took years and years and years of work from many groups, but led, I'd say, by Deng's. Also, took, I think, a lot of knowledge from other groups as well. But in the Deng lab, I think they consolidate all that into a solid, efficient, optimal differentiation protocol for generating islet-like cells from these chemically induced pluripotent stem cells. So these kind of chemically induced islets that had a really high fidelity to the real thing. And then looked at them in primate studies before, show that they're really effective. So that was a key milestone leading to this point. Then they also, and this was a benefit of doing those primate studies, is that they were able to optimize and refine the transplantation strategy. Conventional strategy is you infuse it into the hepatic portal vein, right? But right away, you get large scale graft loss by that, inflammatory response from the blood perfusion. So they developed this alternate method where they transplant to an extrahepatic site underneath the abdominal anterior rectus sheath, and showed that there was superior islet survival and maturation there. And then here is the culmination of it all. They show autologous transplantation of, via this chemically induced pluripotent stem cell intermediate islets into this patient with type 1 diabetes in this exploratory study. And this was a patient with a tough story, diagnosed at 14, roughly, with type 1 diabetes, about 11 years before she entered this trial, 25 years old when she entered the trial, had 2 liver transplants prior to this due to this cryptogenic cirrhosis, probably arising from her life undiagnosed with diabetes, and had 2 liver transplants. In 2014, 2016, had a whole pancreas transplantation that ultimately had to be removed because of severe thrombotic complications. Was already under this immunosuppressive treatment for the maintenance of this second liver transplant. And that's a key point here. So key to mention that the patient was immunosuppressed. But nevertheless, this is a first in-person study of this application. They're out to a year now. They're presenting 1-year results to show feasibility. They do the transplant beneath the abdominal anterior rectus sheath, that approach that they developed. And they show sustained insulin independence 75 days after transplantation, and sustained like solid, right? Time and target glycemic range went from 43% to 96% by 4 months post-transplant. Also, a lot of other indicators of like the long-term systemic glucose levels being at a normal, non-diabetic level. And at a year, she met all the endpoints, the target endpoints, no indication of transplant-related abnormalities. Now, currently maintaining a time and target glycemic range greater than 98%. So, like, cured? I mean, I don't know. This for me is nuts in that there's nothing you can throw a stone at here. They did it. This is a patient chemically induced, autologous. There is an immunosuppression angle, clearly, but I think this is justification for larger scale trials. And really hats off to the Deng group and all the many investigators that contributed to this and many of the foundational studies for this watershed work.
Yeah, pretty incredible publication coming from the Deng lab in China, and really a broad scope of folks were involved with this study, which obviously makes sense if you're focusing on a cell therapy. This is a nice parallel, I suppose, to the work that's already being conducted at Vertex. Of course, those are embryonic stem cell derived islets and are in clinical trials as well. Similarly, Vertex hasn't had a huge publication of their clinical trial. We've seen those N of 1, N of 2 patients that have shown beneficial results or positive results in terms of alleviating the symptoms of type 1 diabetes and restoring insulin secretion and restoring the glycemic index and all that kind of stuff. And this is iPS derived and, importantly, chemically induced iPSCs, which is a really cool advance, and I think very powerful for the reasons that you alluded to in the beginning. Of course, no transgenes, nothing integrating here, just cocktail of chemicals, which kind of almost magically is able to reprogram cells to iPSCs. I mean, you mentioned the limitations. Of course, the immunosuppression is the big one, but really promising. Of course, you got to extend this beyond 1 year. If you really want to call it a quote cure, it's got to be permanent, and a year is, I don't think, enough time to really establish that. There are a couple of other patients that they're looking at too. There's a total of 3 patients in this particular study, but the other 2 patients are apparently not far along yet to actually look at their results. It'll be cool to see the broader scope of the study and see it scaled up, which is inevitably what's going to happen. But Hey, it's a huge medical burden, and nice to see multiple avenues being pursued here.
Yes, I'm glad you mentioned the other two patients because yeah, I mean the question would be why not wait, right? This is a year endpoint. You could wait and get n equals 3. Maybe it's not going as well. I don't know. Cynicism, skepticism. I think the truth is here, it's justified because you got to get this out as quick as possible, right? Because I think that this, months mean lives really in the scope of things. And I think that you need to move to the next stage with these approaches. And this needs to be visible, and this needs to be a foundation to justify a larger scale trial. And also, I think there's some lessons learned here. And specifically, I mean, I love this idea of this anterior rectus sheath, if that's what it's called, this extrahepatic site, because I think that's a model to follow. Uniquely, in the case of treating cell-based treatments for diabetes, I think that this idea of getting it into orthotopic in the pancreas is like not really important and not a prerequisite. So it really validates, I think, a lot of these approaches that have looked at maybe extra hepatic or like microcapsules, or whatever other inserts. And as you said, I mean, you need to go out further than a year about a cure, but I think the time and responsiveness of the patient to this treatment, I think, raises the potential of having a modular approach. I think a lot of patients would be happy to have their, depending on how invasive, some degree of a transplant. And I think those conditions can be optimized to be less invasive. That maybe you have to replace every few years. I think that's close to a cure, and an important milestone. And across the board, I'm really impressed and gratified to see this level of success from this first in human results of a soup to nuts, optimized approach.
Yeah, really cool to see. And you're right. I don't, perhaps it doesn't matter where you put these stem cell derived islets. Like you're saying, it doesn't have to be in the pancreas. As long as you put them in a place that's connected to the bloodstream, the islets are going to do their thing. They're going to secrete the insulin, regulate glucose. I remember Sonia Shrepford did something similar with those hypoimmune islets that she's working on, those like immune shielded islets. I think she put those cells in the muscle, and which obviously doesn't make a lot of sense because if you're talking about the pancreas, but it doesn't matter, right? The point is the cells are connected to the bloodstream, and so the insulin is going to get secreted and the regulation is going to happen that way. So really great stuff happening here from the key laboratory in China. And shifting gears to another study that's coming from China, it's something very different, but is focused on perhaps one of the national animals of China. That's the giant panda, right? That lovable creature that's kind of a hapless giant mammal that is just, everybody falls in love with the panda, right? And perhaps unsurprisingly, the giant panda is endangered, right? It's in trouble. And so there's a lot of work trying to conserve these creatures, protect them, especially within China, and around the world. It's this flagship umbrella species of that country, and it kind of symbolizes biodiversity in a way, right? There's a lot of interest in using traditional assisted reproductive technologies to alleviate this endangered phenomenon, the situation with the giant panda, right? So using different sort of reproductive technologies to help restore the population of the species. But there are issues there, of course, of genetic diversity. You have to not just expand the population, but ideally you want to expand the genetic diversity of the giant pandas. And this is where, who knows, maybe iPSCs can come into play. And this is a concept that we've talked about before on the show, the so-called iPSC zoo, right? We talked about this with Jean Loring, Sebastian Dika, the interest in making iPSCs from endangered species in hopes of one day ultimately making germ cells, sperm and egg, from those iPSCs. We're not there yet, obviously. I remember Sebastian's working on the rhino, and Gene Loring is doing some work with the San Diego Zoo in a comparable way. And here they're trying to do something similar with the giant panda, right? So they're trying to derive iPSCs from the giant panda, period, in hopes of down the road making germ cells to ultimately perhaps restore the population of the panda. It's a big dream. So here they, did the usual thing. They derived primary fibroblasts from the giant panda and generated GP iPSCs, giant panda iPSCs, using a non-integrating episomal vector programming method. So this is not chemical like what the Dang lab's working on. This is a more kind of traditional episomal vector. But they made it work. Long story short, they did the usual characterization of the giant panda iPSCs, the differentiation of the iPSCs into multiple lineages. To culture the cells, they used M-teaser plus, surprise, surprise, to keep them growing. And they're able to show the standard pluripotency networks. These are primed, as you might expect. They're not naive state iPSCs. And also created this species-specific, chemically defined, what they call facile medium, to figure out some of the signaling pathways that are responsible for maintaining pluripotency and actually fostering the proliferation of these giant panda iPSCs. So it's, pretty straightforward study. It's just another study like that's creating iPSCs from an endangered species. It's, providing insights into panda biology, perhaps, but ultimately this is looking towards the future and looking towards the day when you might be able to derive germ cells from these iPSCs from giant panda in hopes of rescuing this very cute and cuddly species.
They are so cute, right? A national treasure in China. And although they're no longer endangered, there are less than 2,000 pandas still in the wild. So this is good and important work. And for me, I love every time one of these stories comes out in the context of species conservation, I get excited because it's another platform for testing the, pluripotent stem cell derived germ cell practicality. Here I go again. I've been outspoken. Don't think it's safe. Just getting on the record again. For humans, I'm saying for humans, I don't think it's safe, but I could be wrong. I've become like really curmudgeonly. I have to be honest. I'm like a real hater on everything nowadays. It can't be done. It can't be done. But as I like to say, it takes a measure of courage to get out there and say that something can't be done, because then you could be proven wrong. All right, Arun, I see you over there like, oh, but it could be done. It could be done. You can only be proven right with it could. No one's ever going to prove you wrong when you're all about could be. All right, so I'm getting out on a limb saying maybe it could be, but it shouldn't be. All right, it's not even about practicality. I'm just an old curmudgeon. Anyway, I digress, but I'll come back to it and say that these conservation efforts could be a great platform. Although I don't know how you vet the product enough over the course. I feel like you would need generations to really see the impact. But still, I mean, you could do sequencing. I think we're getting there technically, that you could do this in this context where there's like a real need, and also get some answers on that front. So I love this. I love this study. I love studies like it. Sebastian Deek, where are you, partner? Get over here. Get some of these cells and get cooking with some of the, the rhino. It didn't work out the first time. I know it's still a lot of possibilities, but the panda's right there.
Panda's right there. And hey, man, that's why we're a good team, because you're a hater and I'm a lover, right? Maybe that works out. But hey, you said they're not endangered anymore, but there's less than 2,000 of them in the wild. Isn't that kind of the definition of endangered? What's the cutoff here?
I don't know. I got on Google there for a second while you were talking. I thought the same exact thing. I mean, what the idea that there's a threshold of endangeredness and then you can go in and out of it is kind of ridiculous. But the panda defies all logic, Arun.
I guess so. I mean, hey, it's a like we talked about, it's a national treasure of China. It's an internationally beloved species. So we want more pandas. Who doesn't want more pandas, right? But I think in addition to just the obvious nature of the study, I think there's some cool biology driving it and behind it of how, how they figured out that these are in the prime state of pluripotency, these iPSCs. They're depending on FGF, activin A signaling for sustained self-renewal, like kind of what's happening in other species as well. And I don't want to gloss over the biology of the study because I think there's a lot of cool stuff in here. There's a lot of interesting angles about conservation of the pluripotency networks across different mammalian species. You've got the usual markers of, SOX2 upregulation, PAL5F1, OCT4, all these kind of things, which aligns very closely with other mammalian species. So maybe there's a deeper study in here somewhere about the conservation of the pluripotency networks. I don't know.
Yeah, no, for sure. I don't want to minimize it. I think there's a lot of science here, and I think you got to make the conservation sell kind of, I guess, to get it. It's a science paper. I mean, it's a high caliber, so I think you need to touch all the bases there. But yeah, there's a lot there, and the effort alone should be applauded. So good show. Moving on to the humans, 'cause I'm trying to conserve the humans over here, Arun, you people caring about nature. Well, not me. I'm talking about a paper about humans in this case, and really a cool concept here. And this, I just have to say at this point, we've already recorded our interview with Alfonso Arias. So this roundup here is part of Behzad's show, but we had such a great interview. You guys got to listen in a couple weeks to that one, because we talked a lot about these human models, embryo models, and the foundational work from people like Nicolas Rivron, who's one of the lead authors on this. And it was such a great interview. You guys are going to love it. Stay tuned for that. But getting back to this show, this is a story that illustrates or kind of elucidates, I should say, some of the underlying potential of human embryos for diapause. Okay, diapause is this cool thing. I mean, you guys might remember it from your, college or graduate developmental biology, but it's a lot of mammalian species have this capacity to temporarily delay development. I've mostly been aware of it in mice is the model that we talk about it because that's our model, right? But a lot of animals, over 130 mammalian species can do this. It's this dormancy, embryonic diapause, that can be triggered by a variety of factors in a species-specific manner, but it's mediated by these extrinsic hormonal signals and changes. But the embryo remains responsive to signals that can then restart, kickstart. It's this reactivation. And this is what's really cool that I didn't know, is that these interspecies uterine transfer experiments have shown that the capacity for diapause may be retained in embryos, even if they aren't capable of it. Like humans, for example, aren't capable of diapause. We don't have that as a part of our reproductive biology, and neither do sheep. Sheep blastocysts, they don't naturally enter diapause, but they can enter diapause if you put them into a mouse uterus and induce diapause by those extrinsic hormone signals. So there seems to be this kind of latent capacity that may be maintained across the board or on a species-specific level, but it raises a question of like, can human, even though we don't have that a part of our biology now, and the totality of it, is the embryo, embryo intrinsic capacity for diapause still in play? And this study used blastoids. So we're going to talk about that next episode with Alfonso. But in this study, they used blastoids, these kind of not rudimentary, but basic or first step embryoid models in human in this case. That's the part of Nicolas Rivron, who's one of the co-leads here, and also Aydin Bulut Karslioglu, who previously, he's the co-lead here, who previously showed in the Romulo Santos lab that mTOR pathway, specifically mTOR inhibition, could induce diapause in mouse blastocysts for several weeks. And importantly, that dormant state is reversible. You can take those reactivated embryos after weeks, Arun, and then they give rise to live fertile mice. So this is crazy. and like the reproductive biology, reproductive medicine potential for this is pretty clear in terms of like pacing, and that's the part of the title of this is pacing of human embryos for IVF. That'd be a really important innovation. Here, they use the similar approach. They use this moderate inhibition of mTOR signaling in human blastoids to induce, both in blastoids and in human pluripotent stem cells, to induce this kind of diapause, which had a lot of facets. They showed they didn't proliferate, limited the developmental progression in terms of like differentiation. They didn't attach to these endometrial outgrowth cells. So I thought this was a really cool. I love diapause just as a concept. It's really cool. I think this is also a great illustration of the value of these embryo models, right? Because this is something you just wouldn't be able to do in the human concept. I mean, yeah, sure, you could get donated embryos, but the scale, I think, you need to really interrogate this. You couldn't have done with that embryo models. So I think this is a real indicator of the tools of our times, Arun, and how they have some real deep impact, not just mechanistically in terms of understanding concepts like diapause, but maybe their application in a practical way to improve IVF or some other element of embryo development, organoids, pacing of development across the board, and differentiation, I think, is a key concept that needs to be dissected. So an important study here in Cell article. Great story using these human embryo models to their potential.
Yeah, absolutely. And like what you're saying, I think this is a study that perfectly exemplifies the power of these models, because I think this is a study that really could only be done using blastoids and these embryo models, because you just need so many of these things to unravel the mechanism behind diapause and the open the black box of development in this way. This is the perfect application. And yeah, this is great timing. Of course, we have Alfonso Martinez Arias on the show, and not too long from now. Really beautiful study, a lot of basic science, awesome basic science here, but also potentially implications for IVF, like what you alluded to. One of the things to discuss here, of course, is mTOR, this central signaling pathway, which has a lot of roles in cancer and other applications of as well, of course. And they say it themselves. This is probably only one of the components involved in dormancy regulation. There's, even though they're showing mTOR inhibition is sufficient to trigger dormancy, these dormancy cultured systems might be improved by the introduction of modulating other pathways, right? Like they alluded to that so with some of the mouse embryo work. So I think there's a lot of other work that has to be done to follow up the study, but a really beautiful application of human embryo model technology.
Yeah, for sure. I mean, mTOR is a sledgehammer, right? It's one of those things where if mTOR does it, then you got to drill a little deeper to get some specificity, because mTOR does a lot of things. You might be a little wary of mucking around with mTOR at the founder stage of embryogenesis, right? But yeah, like you said, I think an important application, and I just want to highlight also the time scale of scientific progress here for a minute, because like that mTOR demonstration, mouse embryos, which was an amazing step forward at the time, a big Nature paper. That was 2016. Here we are, a little less than a decade later, right? And it took the introduction of the blastoid models, I think, as a prerequisite. But then those ideas come together. Blastoids have been around for some time. And now here, I mean, it took years, right, to get to this point where the questions were asked and answered. And you look at the last summer with the human embryo models really taking off in terms of like the progression that we're able to approximate here. It makes me think, with the excitement, anticipation about what's coming down the pike in terms of looking at these more complex events at scale, and incorporating all these tools to really to unravel. I mean, this is this, I think, study, as you said, it could only be done. You would only be done, I think, at this level, because it's such a primary early event, right? You're stopping the progression. It's going to be a lot more complex as you incorporate all the cellular morphogenesis, et cetera, and the more advanced embryos. But I think those questions are being answered or asked at least and addressed. And it's a really exciting time, as we say every single day. It seems like such an exciting time. Peak stem cells, it seems like, will never end.
Well, you know what's coming next, Dylan? It's the derivation of germ cells.
Oh boy.
Stay tuned. I know. I'm just kidding, man. I just wanted to see your response there.
I'm dying
Um,
inside.
I know. Moving on to our last paper of the roundup. This is actually a Nature Genetics article. We don't discuss Nature Genetics too much here on the show because they don't have a lot of, I guess, organoid or stem cell directly relevant stories, but of course, extremely well regarded journal in the field. Here they're talking about an organoid-ish system that's used for sustaining epithelial cultures for a long time. They call them epithelioids, but they're not really oids. They're not really three-dimensional organoids, but we can talk about it. So this is a Nature Genetics paper titled Self-Sustaining Long-Term 3D Epithelioid Cultures that reveal drivers of clonal expansion in the esophageal epithelium. And there's a big CRISPR screen and big screen towards the end of this paper, which is, I guess why it ultimately ended up in Nature Genetics. So this is coming from the lab of Philip Jones at the University of Cambridge. First author here is Albert Herms. And this is developing a new model to study epithelial culture and epithelial mutations in particular. So aging epithelia they accumulate somatic mutations. The skin accumulates mutations a lot. Because the skin divides a lot. These cultures of epithelia, aging epithelia, are subjected to selection, influenced by, of course, its intrinsic genetic factors, but also the environment. The skin's exposed to so many of different stimuli, and that can induce mutations for sure. The skin cancer comes to mind, of course, right? There's apparently not a lot of great culture systems to show the study of aging epithelia and their accumulation of mutations, and their other associated long-term biological processes. So here they describe these, what they call epithelioids, which is an easy, cost-effective method of actually growing a bunch of different types of human and mouse epithelia. So they compare these two organoids and demonstrate their advantages. So first, they derived esophageal epithelioids, which are able to self-maintain, get this, without passaging for at least a year, which is insane to me because epithelial cultures are probably some of the fastest dividing cells out there. And you're telling me you can grow these things without passaging, and I think they said just a couple of media changes a week for a whole year. I think that's going to make a lot of people happy. And they maintained a three-dimensional structure. It's not an organoid, like I told you. It's more of like a thick sheet, this three-dimensional thick sheet with a proliferative basal cells that actually differentiate into the suprabasal cells, ultimately that shed and retain their genomic stability. That's important. So it's almost like a pseudo skin, like a very thin layer of skin that they created in this, almost like a transwell system that importantly retains its genomic stability, but you can maintain it without passaging for a year. So maybe that's part of the why, like the reason why they have the genomic stability, because these cells aren't maybe dividing as much. I don't know. Anyways, they did a ton of live imaging over 5 months showing that the epithelioids in some aspects can replicate in vivo cell dynamics of migration and the proper structures here as well. And then they brought the nature genetics part of the story into the fray, where they introduced a lot of genetic manipulation in CRISPR screens, right, to actually enable the study of how cells compete in this epithelioid environment, how selection happens in this artificial system, and how to show how you could use an anti-cancer treatment to actually modulate competition between cancerous transformed epithelioid cell types and also wild type cells. And they wrap things up, of course, like I said, with a targeted CRISPR screen to show that epithelioids can recapitulate mutant gene selection an aging human esophagus and actually identify additional drivers of clonal expansion. So it's a nice way to figure out some of the genetic networks that are underpinning the fitness of different cell types in these populations. Really neat system. I was just, like I said, blown away by the fact that they can maintain these things without passaging for a year, but still replicate some of the architecture that's actually found in the skin. And like the other part of this is not just a mouse study. They derived epithelioids from multiple tissue types, bladder, esophagus, in mouse and in human as well. So it seems like a really useful system. These are kind of my favorite studies to develop a model and then characterize that model across a bunch of different applications. I think it's going to be widely used in this field.
I am buying the oidy of these things, and it's for the grounds that you stated. I mean, I like you think of more 3D, but I think the complexity or the stability more, right? Because it's also not an organoid. It doesn't have like multiple different cell types, but it's exactly what it says. It's a stable epithelial oid. It's like ish. Has the that in vivo, I think, component, multiple components. And more than anything, as you said, it's just an amazing tool. I think pharma is going to love this. Something stable like that goes out to a year, get it from multiple cell types, human. I mean, I think this is a big win. And the genetic stuff, I think, is just to hit you over the head and get it in, right? Of course, but to hit you over the head with how you can apply this thing and combine it with the modern day genomics to like a sweeping way to ask big questions. So great study, great tool, as you said. A lot of people are going to be uh, sending notes to the Jones lab asking for the recipe.
Yeah, and that's the key. It looks so simple, right? It just looks like a transwell that is available in most labs, and they see that, yeah, this is kind of the key to making these things survive long term. And of course, there's certain culture conditions that they have to incorporate here as well. But I'm a fan of those simple studies. Like a couple of years ago, Sean Wu, my former grad school advisor, who was on the show like a couple years ago, I guess, published this really great paper in Cell Stem Cell where they were able to cause massive proliferation of iPS cardiomyocytes, which normally don't divide, just by hitting them with the perfect amount of cheer. Cheer, small molecule that we use all over in our cell cultures. And I wish I was the one to think of it because it was so simple, right? And similarly, this is it seems like a pretty simple approach to something that could have a lot of impact. It's highly scalable. You can manipulate it with genetics. And like you're alluding to, I'm sure pharma is going to be all over this.
Yes, primary cells and their application, full circle since the days of Hans Clevers and the intestinal organoids. Now thought we were out of oids. Well, here you go. We could talk a little bit about that. Primary cells, adult stem cells, etc., with our guest, uh, Behzad Yeganeh, in a minute. But first, I got a quick message from STEMCELL Technologies. Are you modeling human airway in vitro? Are your cultures physiologically relevant? Can you generate these cultures consistently? Learn more about Pneumacult, offered by STEMCELL Technologies. It's a serum and bovine pituitary extract free cell culture media for human airway epithelial cells. Expand these cells for extended passages while maintaining air-liquid interface differentiation potential to study respiratory biology, infections, and drug responses. Explore more at www.stemcell.com/pneumacult. All right, you guys, today on the show, we have with us a special guest from the Children's Hospital of Eastern Ontario Research Institute and the University of Ottawa, where he is Associate Scientist, Dr. Behzad Yeganeh. Dr. Yeganeh is a developmental cell biologist with research experience and expertise in lung development, neonatal lung diseases, and lung injury. His current research interests include understanding the mechanisms of lung development and neonatal lung diseases, injury, and repair. In addition, he has an active interest in uncovering the molecular mechanisms regulating autophagy and apoptosis to develop new methods and therapeutic strategies, including regenerative medicine and preventative treatments. Dr. Yeganeh, thank you so much again for joining us today on the show.
Oh, thank you very much for having me for such an interesting podcast.
So yes, thanks so much for joining us, Behzad. It's always great to chat with folks who have been listeners of the show and have had interest in our our conversations. I mean, your work revolves on lung development, lung diseases, as well as the mechanisms of autophagy and apoptosis. But just to start things off, why don't you give us an overview of, in your own words, of what your research is all about, and maybe your road to becoming a stem cell biologist in the first place?
Yeah, thank you very much, Aaron. I don't call myself a stem cell biologist. This is, a portion of my work. But looking at my research and my career on science, I did my PhD in cancer biology in the University of Manitoba in Canada. And for postdoc training, I moved to Toronto at SickKids Research Hospital when my research was more focused on developmental biology, particularly on lung development and premature related lung disease. So these the different area of the research actually helped me to become more interested, the mechanism of the cell signaling, which are related in the stem cell biology, because part of the work in the lab that I was working at SickKids, they were doing the stem cell work as well. So that was the initial time that I got exposed to the stem cell work as well. As as we were also doing the lung injury and finding a therapeutic approach for treatment of the lung injury, particularly in the neonatal premature babies. That was the time point.
Yeah, I mean, I feel like maybe in this newer generation, there are just true, born and bred stem cell biologists. But I feel like at the root, we're all in part developmental biologists, right? Who have come upon stem cell models that we can use as tools, or ultimately, I mean, even therapies. And that's what's I think so exciting about being a developmental biologist or having an interest in any point in, even neonatal stages of development, organs in flux, let's call it. It's an exciting time for all of us who appreciate that, I think, because we have access, right, to more and more sophisticated models for interrogating those processes. But also the clinical implications and applications of our science and our interest are also really expanding in scope. We talk a lot about cells, tissues, organs that can be modeled using stem cells on this show, but I would argue that the lung is not really as well represented, particularly relative to the unmet healthcare need of diseases that affect the lung. Both in adulthood as well as like neonatally, right? And premature babies, et cetera. What's the state of the art for regenerative and cell-based? And I know you're not a stem cell biologist, so I mean, you take free reign here with this question, but I would ask, for our audience, for myself, what's like the state of the art of regenerative, cell-based, whatever therapy for the lung? And what are the greatest obstacles right now to advancing and or expanding these approaches, either to a more general audience, more broadly, or to increase the efficacy of those approaches?
Yeah, lung is one of the most sophisticated and complex organ in the body. And because of the nature of the lung, the therapeutic approach, particularly when it comes to the cell therapy, it's so difficult, because the physiology of the lung at the distal area compared to the proximal area, it is totally different. Most of the lung injury, no matter what, is happening at the alveolar area, the distal area, which are exposed to the air. Specifically for the premature babies, because the lung is not fully developed, they're undergoing different interventions, particularly mechanical ventilation and also hyperoxia, which is good. These approaches are life-saving for premature babies, and we have quite progress in the last, I would say 2 decades or 3 decades. But the problem is that all these interventions also causing epithelium layer damage or lung injury, which eventually this stays in these patients, even when they are adult. And there is a terminology called bronchopulmonary dysplasia, which the functionality of lung is actually altered, and they are not fully functional. These patients, although they are survived and they are healthy, but the lung is not fully functional. So it's very important to improve the lung injury at the very earliest stage when they are just born or they are undergoing this therapeutic mechanical ventilation or hyperoxia treatment. At the moment, actually, one of the major progress is the cell therapy using mesenchymal stem cells from cord blood or from bone marrow. And I think there are clinical trials, even here in Ottawa, investigators, they are using mesenchymal stem cells for therapeutic purpose. Coming to the iPS cells, there is not much progress, and it comes mostly because of the complexity of the lung itself and how we can drive the iPS specifically for the, let's say, bring them to the alveolar type 2 cells or specific cells. It's very difficult. And so far, we didn't have a conclusive answers.
Yeah, so actually, let's sort of stay in that same realm. You had a recent paper where you talked about generating these adult multipotent stem cell-like cells from somatic tissues, maybe even inspired a little bit by the work we're talking about with iPSCs and fate reprogramming that ultimately led to induced pluripotent stem cells. I know it's a different cell type, obviously, but one interesting takeaway here is that this is a non-transgenic suspension-induced method of inducing fate transition from to these adult stem cell-like cells. And we actually talk about fate plasticity a lot on the show and how things can go back and forth, perhaps more often than we would expect in the human body. So maybe this is another example of that, of fate plasticity. Could you talk a little bit more about this work and maybe what it tells us about fate plasticity in somatic cells?
Sure. We know that cell therapy or iPS, since it's introduced in 2006, which is almost 18 years, created a lot of hope for treatment for the cell therapy. But in reality, we are facing a lot of challenges. We know that there are difficulties on tumorigenicity, genetic instability, and adult stem cell or tissue stem cells are already in place. These are more commitment cells, and all those challenges which are facing with iPS or embryonic stem cells in general, we don't have it. But the problem with the adult stem cells or tissue-specific stem cells is that we cannot find them in the high quantity. I mean, cord blood is a good source, or bone marrow, but the rest of the tissue, let's say from liver or from muscles or other organs, it's very difficult. The population is very rare. So I think it's a good investment to put more time and also resources on to develop a new techniques or method to be able to increase the quantification of the tissue stem cells. So this work is trying to address these questions using of the adult stem cells, introducing a new method. Coming to this paper study, I have to elaborate a little bit more about the cell biology, because their audience are biologists and scientists. Most of the cells coming from the solid tissue, as we culture them in the plate, they are attached dependent. They have to attach to the plate and to grow. And this is the nature of the cells. Even in the reality, in the body, cells, they need the cell-cell contact and also cell basement contact to survive and grow. And if you detach them from plate, they won't survive. They undergo a process of apoptotic cell death, which we call it detachment-induced cell death. It's a terminology called anoic cell death. I had an observation years ago when I was completing my PhD, because I was looking for cancer stem cells on my model of lung adenocarcinoma. And by accident, I noticed that the normal cells, once they are placed on the not adhesive plates and they are not able to attach, they are floating on the media. And next day you will find them, they are undergoing cell death. But by accident, I noticed that at this time they are expressing a stem cell markers, although they die because of the nature of their living. I took this observation in more deep investigation, and I found that why, because at that time, apoptosis was not my area of the research. That's why I changed my direction. I did some experience working on apoptosis to identify that, what exactly happening to these cells. At that time, I was using fibroblasts as a model from mouse. And so I decided to force these cells to contact each other using gravity, because at that time, we didn't have a plate, a U-bottom plate, a 96-well plate. And based on the gravity, the cells were forced to attach each other and survive and make spheroid. And then I harvest these small spheroids and characterize them. And I noticed that, yes, they are expressing, for example, in fibroblast case, they were expressing mesenchyme stem cell markers. When I found my position here, I started to have a deeper investigation in this phenomenon and find out if it's something universal or not. Is just related to the cells from mesoderm, or is something happening, in the cells which are coming from ectoderm or from endoderm, and how about human cells as well? So to my surprise, I noticed that yes, we can keep these cells alive. They grow and they make spheroids, and the spheroids are having the characteristic of their own adult or tissue-specific stem cells. Let's say, for example, the skin or dermal fibroblasts, they are coming from mesenchyme cells, which are mesoderm origin. You can find in this study that once you characterize this, you can see them, no matter these are mouse fibroblasts coming from the skin or human fibroblasts coming from the skin, they have the characteristic of the mesenchyme stem cells. Similarly, I used cells from ectoderm origin. I used keratinocytes, and I found that based on the current knowledge that we have on the skin stem cells, once you have them on the spheroid, once you have them in suspension culture and push them to become and create the spheroids, they have the keratinocyte stem cell characteristic. For endoderm, I use the cells from liver hepatocytes, and same phenomenon I observe. I think I don't know exactly what is happening in terms of the mechanism, but whatever it is, it's a small steps forward to find or to improve the technique or method to get more tissue-specific stem cells for therapeutic applications.
Yeah, I'm with you on that. I mean, of course, I appreciate the amazing promise of pluripotent stem cells, iPS cells for modeling and therapy, everything. But, the adult stem cells there are autologous in many cases, right there for the taking if you can figure out a way to leverage them, deploy them, expand them, maintain them, whatever you want to call it. There's a lot of obstacles, as you're alluding to there, to refining it to, ready for prime time. But the concept is old, not old, like stale. Like it's not an original idea, because it's long been since, bone marrow transplant, right? It's been out there and it's worked. So why not? And the idea of the multipotent adult stem cells has a long history, but also a bit of a fraught history, right? When I first it was in my early training days, there was the idea of the circulating multipotent adult stem cells from Catherine Verfaillie at Minnesota, which were controversial. I saw, looking into it before the interview just a few months ago in June, that seminal Nature paper that described them was retracted finally after 22 years. Then there was this kind of scandalous claims of Pierre Anversa from Harvard suggesting that bone marrow-derived MSCs had regenerative potential in the heart. Not exaggerating, over 100 million, probably hundreds of millions of NIH money sunk on that premise. Then more recently, there was the STAP controversy, where bogus claims of these so-called stimulus-triggered acquisition of pluripotency cells ultimately led to suicide of Yoshiki Sasai, right? So not just like fraud, but dramatic history here concerning these multipotent adult stem cells, reflecting, I think, that this collective longing we have for a somatic cell that has true regenerative potential, but so far elusive, I would say, putting it generously. What do you think are the limits in terms of the cellular purview of specifically your suspension-induced stem cells? Is it like tissue specific? You spoke of the different cell types of origin from which they came, from whence they came. Is there kind of like a narrow windows of potency there? Second question there is, like, do you think you can get a truly pluripotent cell from these somatic cells?
That's a very good question. I mean, I don't have a proper answer, because the study that we ran and we published, it was just starting, just beginning. And because of the limitation of the financial source, we were not able to fully and functionally characterize these cells. But we did RNA sequencing, and we found that once this population, a small portion, almost 10% of the cells in the mouse fibroblast dermal skin of the mouse, once they are making spheroid, almost 10%, they have a stem cell property. I mean, they have those genetic characteristics. In terms of the functionality, that would be great to run experiment to see, how functional these cells are and how these cells are able to regenerate if there is a damage, or in different concept to see that. I think these are fundamental questions to address the functionality of these cells. Also, what we just did, we characterized these cells based on the current knowledge that we have, on mesenchymal stem cells or keratinocyte stem cells, or let's say stem cells from liver, what markers they are expressing. If these cells are functional to heal regenerated liver or regenerated tissue, they need to be actually examined for sure.
Yeah, we're always interested in ways to improve the current biomanufacturing processes associated with cell therapies. I mean, we've had great conversations in the past with folks at, say, Blue Rock Therapeutics and other places as well about, what are the appropriate quality control steps for cell manufacturing if you're going to apply these cells for cell therapy, like, what we're alluding to here. And I mean, we talked about it a little bit, right? You have this interest in methods to actually address the risk of, say, tumorigenicity, genetic and epigenetic instability, challenges of scalability and manufacturing. And these are really important topics that anybody who wants to make cells for cell therapy we have to address these topics. And in the current age of iPS-based cell therapies, so many of which are in clinical trials right now, these are just as important. QC is just so critical. Quality control is just so critical to making sure that safety, the high bar for safety, is met in whatever therapy you're hoping to develop. So tell us more about your interest in this particular area of addressing these challenges of cell-based manufacturing.
That's actually quite a very interesting topic, Aaron. Coming to IPS, we know that all these challenges, you just actually mentioned that. And one of the paper that I was interested to talk about it a little bit was related to a group in MIT that they published to address this big question, how they can reduce the number of the cells with the tumorigenicity capacity on iPS cell. So the title of the paper they published is Label-Free and High Throughput Removal of the Residual Undifferentiated Cells from iPS-derived spinal cord progenitor cells. So based on the method that they introduced, they were able to get rid of the majority of the tumoric cells in this using their method. But they claim that they were able to get almost 98% purity of the cells, which they are not tumorigenic. 98% is a good number when at first we hear and first we see it, but in reality, when it's come to the quality control, as you mentioned, even 2% is so high. Even 1 in a million is very high. So there are efforts on iPS cells to overcome and answer the tumorigenicity of these iPS cells. But to me, I think we are very far coming to the clinic, coming to the real transplantation. And all the clinical trial, as you mentioned, I could call them, they are in the phase 1 or phase 2, and is mostly experimental stage. Having said that, although it's a good step, but this paper actually is highlighting this big question at the same time, that although we are trying to answer this pitfall and this major concern, but this is reality. This is there, and even 98% is so far from the reality of transplantation of PS cells. And then when I went to this recent conference in Hamburg, and I noticed that majority of the talk and also posters, still they are dealing and talking about the iPS cells, and not much and not too many cells or posters related to tissue-specific stem cells or tissue adult stem cells. So one of the reasons that I wanted to participate in this podcast to bring this conversation and highlighted that the adult stem cells are already there. They are established. They are safe. They don't have much concern, but not much work is going in the past is done by investigators. Yes, we have work related to the bone marrow or cord blood, but coming to the in general idea of the tissue stem cells, there is not much interest. And I think it's good to bring the attention of the scientists that to improve the technique or methods to increase the quantity of the tissue-specific stem cells, because the quantity of the tissue-specific stem cells, the only problem is we cannot find them in high quantity. If any technique, any method can actually boost up the number, there is a new door open for cell therapy, safe cell therapy. It's more diversity, not just cord blood or not just bone marrow cells.
Yeah, I agree with that. I think that, as I said before, it's a long and fraught history, and there's a bit of a stain on MSCs because of a lot of unsavory actors, some of whom I mentioned. But I think it's making a comeback. And I think the key there really is characterization, right? And mechanism. Just last episode, we covered this story from Rubin Lab, where they described novel means of expanding the true satellite cells from primary muscle biopsy. And I think that was the key, right? We know they're there. We know they're scarce. We need to have a well-characterized and mechanistically interrogated pathway before we can comfortably inject these cells. And, I think we're on our way. MSC's making a comeback, making a play, although I would agree with you. iPS cells, I think, have certainly taken the spotlight for the most part. I want to circle back to your autophagy studies, deciphering fundamental mechanisms that underlie autophagy that resulted in a Nobel Prize to Yoshinori Asumi in 2016, although a lot of argument that there should have been wider recognition. A lot of other players in that process. His work primarily was in yeast, but the importance of autophagy in multiple aspects of human health and disease continue to this day to be elucidated, in part by your own group. Can you elaborate a bit on your work linking autophagy to healthy lung development? That's where your focus is. Are there implications there for regenerative approaches to lung disease and or injury downstream of that, mechanical ventilation or hyperoxia?
Yeah, thank you very much for bringing up this. When I finished my PhD on cancer biology, and after the observation that I had, I switched my attention to the cell death pathways. And of course, autophagy was part of the cell death pathway. Autophagy is, I would call it, is not cell death response. It's mostly there to help cells to survive. But if it fails, eventually undergo cell death. It goes to the apoptotic cell death or other form of the cell death, for sure. So when I joined the lab at SickKids, one of the line of my work was based on the preliminary data I had, was to look at the role of the autophagy and also cell death pathways on lung development. And these pathways basically are important because all the injury that we have also related to the premature babies are directly connected to these pathways. So we found that the process of the apoptosis, which is fundamental for all the developmental tissue, brain or kidney, and similarly in the lung, cell death apoptosis also important to make sure that we have a proper lung development. And what is actually orchestrating and governing this, it seems it's autophagy. Autophagy is controlling the induction of the apoptotic pathway at the two different stages of the lung development, specifically in the mouse model, which is so resemble to human lung development. And we predict that, in the condition like bronchopulmonary dysplasia or in premature babies, they are suffering not proper, fully developed lung development. These pathways are somehow targeted. There is a potential to use these pathways, target them for therapeutic approach as well. But we are very far about that. I mean, it needs a rigorous investigation and also very sophisticated therapeutic approach on animal model to overcome if there is any, injury because of the early and prematurity of the lung, for sure.
Yeah, I think it's something we allude to a lot here on the show is no matter what cell type you're working in, if you have an eye towards the clinic, you have to make sure that your science, the foundational science, is sound, right? Whether you're working with adult stem cell populations, induced pluripotent stem cell derived cells, the last thing we want is, of course, another black eye for the field. And unfortunately, the field has had a number of these in the past. But I like to think that, as a whole, we're progressing in the right direction, learning from our mistakes and really taking that rigorous quality control, like what we talked about, the rigorous quality control to make sure that ultimately the patients who receive these technologies will benefit from them and not suffer as a result of these therapies. So thank you so much for your perspective, Behzad. It's been a pleasure to chat with you on this topic. We're going to let you go, but before we do, we got a couple of peripheral questions that we'd love to ask our listeners. And you gave some great answers to some of these, so we want to give you a shot to answer some of these. So first, if you could answer any single scientific question, regardless of your expertise or chosen field, what would it be?
One of the major question, the big question is, how and why, the life started and emerged on our planet Earth. Is it really started here on Earth, or is coming from outside? Is it really a mystery and big questions for me?
Yeah, I would agree. It's one of my favorite questions. I would argue one of the, if perhaps the most important question of all is, are we alone and where do we come from, right?
That's correct. Yeah.
Yeah. And finally, what's the biggest misconception about science that you'd like to resolve?
Yeah, I think most people, particularly scientists, even young scientists, they think that science is absolute, and the scientists with the high record or giants, they are always right. And I think this is something that we already suffered a lot in the past, as Dejan also mentioned about a couple of them, for example, the stem cells, or in the history, we have quite many, I can call many, that it's basically coming because the young scientists, they were not able to speak up and challenge them for example, for stem cells, everybody was believing and trusting what the Vacanti group or SASI group, they were claiming, although it was misconduct, although it was false data. Well, we have in the history of science a lot. When you are, let's say, if you are sitting in a room which old and big scientists sitting there and claiming something which is completely extraordinary, but you think that it doesn't make sense to you. I mean, most of the time we don't dare to challenge. I don't know. I mean, this is something that it's, to me, is one of the big misconception that we have in science.
Yeah, absolutely. I mean, and sometimes there's some bad actors and fraud. Sometimes you're just wrong, right? Or you misapprehension of the data, conclusions that are not necessarily supported, but not in conflict with the observation. So, I mean, I think that it's a key element of science, and the method is to revisit all these old questions. All truths are provisional, right? Theorems, not facts. And I think the work that you continue to do in search of this long-sought adult stem cell and expanding our understanding of what the actual biological purview and potency of that cell are really key. We need to keep that torch in play, and we're grateful that we've got you to carry it amongst many other researchers who are looking into this question. Thank you so much for sharing your experience there, your insight, and your story of your path to this point. Behzad, really appreciate you joining us. All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or via email at info@stemcellpodcast.com with feedback or to suggest guests.